Matt's got to share his presentation with us now, hopefully.
Yeah.
Good afternoon, Matt. Thank you.
Good afternoon. I can't see my camera. Maybe you can see it.
We can see you, yeah. Sometimes.
Oh, perfect. Very good.
We can see you fine.
Share screen. Sharing.
Okay, so just to let you know, while Matt's doing that, an introduction to Matt. Matt is one of our pathologists, NIAB pathologists at East Malling, and he's been doing a lot of work in recent years on apple canker. We did hear a little bit earlier today, Matt, from Robert Saville about the Growing Kent Medway funded project on canker, which I think you'll be talking about this afternoon. So
yeah, I'll reference it every now and then.
It's I don't want to put any pressure on you, but it's the topic that's always of most interest to the vast majority of apple growers out there. So I'm sure they'll be delighted to hear what you've got to say.
Okay, thank you. Yeah, so I'm a Matt Papp-Rupar, and I have a researcher here at NIAB BMR now for about 4 or 5 years. So most of you will already know me. And I decided that I'm on every beginning of every talk from now, and I'm going to put just the basic of apple canker, just to bring on the challenges and what you already can do. So caused by Neonectia altissima, it's a fungal disease, and the first thing that's really important, it's infecting only wounds on the trees. So there'll be petal scar, leaf scars, or pruning and picking wound, and they are available all year round in different amounts and different times of year. And the spores with which the fungus infects with Neonectia vets are also available all year round. So it needs all year round control, really. You have conidia produced on very young cankers from this year's, and then they get rain splash dispersed within the tree or between the neighboring trees. But then the ascospores on the old cankers, like 3 months and older, they get wind dispersed from an old orchard to a new orchard. So it's the hygiene is really important, the removal of inoculum, especially in the young plantations where if the canker is on the main stem, you can kill up to 30% of the trees, sometimes in the first 2 or 3 years where the new orchard is planted. And that is mainly because most of the cultivars that we're growing, such as your galas and Braeburns and the rest, are extremely susceptible. And to the point where you get canker in very early on, you get reduction of yield, you get dead trees, and then reduction of quality of the fruit when the orchard matures. So the disease is quite difficult to control because we're lacking a lot of effective chemical controls now. With the copper was really effective, is now completely gone. We have tabiconazole, Captain is being removed in the next couple of years. And it's really difficult to protect those leaf scars really well in different conditions, such as at leaf fall. And recently, with all the climate change that we're getting a lot of waterlogging and very warm winters in which fungus can sporulate really happily. And then we get stress in the summer as well with droughts and heat stress, extra stressing the tree, making them potentially even more susceptible, is sort of compounding all this. So the canker management as is, I already glanced over the fact that there's very little in terms of chemical control. So the most important thing that you can do at the moment is removal of inoculum, which is quite expensive and labor consuming. So that's your bottleneck if you have enough labor. And ideally you would remove inoculum 2 to 3 times a year. And especially it was found effective in Netherlands, Germany, and France. They saw that summer before harvest pruning, before leaf fall, pruning of the cankers are really decreased. So if you have just enough staff to do 1 extra canker pruning, that would be summer before, just before you go and pick and before the leaf scar. You reduce inoculum there, and it's really important. And another thing that should really be that all the canker should be really removed from the orchard, either mulched, ground up, or completely removed, maybe turned into either composted somewhere, or because they will produce, if they're not completely destroyed, they will produce ascospores and keep inoculating trees even when the wood is on the ground. One last thing of canker management that you can already do is nutrition. We know that a really high nitrogen, either as a foliar application or soil application, will really, really increase your canker susceptibility of the trees. Even with such little as 50 kilograms of urea applied to the soil per hectare, you significantly increase canker susceptibility. So with that sort of overview, we're going to just quickly go through some of our canker management, what we're doing in terms of research at East Malling. So in very short term, we're going to look at efficacy trials for existing products that you might be able to use. And then what we're doing in the novel microbial amendments, such as new biocontrols that we're trying to develop. Then we're going to look a bit of a soil and site research that we've done, and then what we're doing in the soil amendments for improved resilience, not just to canker, but to other things as well. So first, we're going to quickly go. We have 2 ongoing trials to find new products to control canker, particularly at leaf fall. And we have AHDB funded trial, and we have the Growing Kent and Medway Resilient Apple Orchard trial, and they sort of go hand in hand. Both looked at new products that are already on the market. The AHDB trial really focuses on things that maybe just need an EMU and they might be available for use, while in the Resilient Apple Orchard trials, we're looking at a bit more holistically. We're looking at the defense induction products as well. We're looking at biostimulants together with biocontrols and together with nutrition manipulation, and we're trying to put together some programs. So we have 2 big trials here, and we expect the first results this summer, and then we're going to continue the next. So this season, we're going to continue these trials with the best products, and to just get a repeat, so we can help with the application for EMUs or for registration of new products. So that is in the very short term. But then we're looking for the new microbial amendments as well, or new biocontrol, which so far wasn't very successful. Everything that we have tried commercially wasn't very effective, at least not on its own. And that was tested here and in New Zealand, and other people that they just don't have enough. We don't have very good biocontrol efficacy against apple canker. But here at NIAB, we, for the last 5 years, have focused on apple endophytes as a novel strategy. And these are fungi and bacteria that actually live in the tree rather than on the surface of the tree. So when you have a wound created by leaf dropping off or a picking wound, that endophyte we hope would be already in to protect the tree from within, if you wish. So we've done in the previous few projects we found a fungal endophyte, Epicoccum nigrum, which was much more abundant and resistant than in susceptible cultivars. That was the first hint. And then we've run some field trials in which was quite successful when you put Epicoccum spores on the pruning wounds or leaf scars before Neonectria spores. We got about 50% reduction of infection rate, but it didn't really colonize apple in the long run. So even though it was found as an endophyte living within the tree, it's not very, very good colonizer. The good thing, though, it was that it didn't cause any other symptoms, and it has potential to control more than just apple canker. We know that it does, at least in the lab, control polar rot, so Phytophthora cactorum in apple as well, and it could control other things. And we haven't tested it, but we hope in the near future we'll be able to. Then we looked at what other things we can find apart from Epicoccum, and we started looking at bacteria as well. And we found that actually, when you look at apple trees across different seasons and what is residing in, what kind of bacteria and fungi are living in, we found that actually bacterial communities might be, or bacterial biocontrol might be better than fungal, because they seem to be more stable in terms of bacterial community sizes and composition, seem to be more stable from spring to summer to autumn to spring and across seasons. So we found, by looking at across different sites in Kent and across two different seasons, we found this Xiangming bacteria, which were really highly abundant in resistant compared to susceptible cultivars again. And they also, in our trial on our site, their abundance, so the more abundant Xiangming bacteria were, the higher the canopy health was of the trees, so which is sort of connected to canker. The less canker you have in the more canopy, the healthier the canopy it is. And we know from literature that Xiangming does promote plant growth and can increase tolerance to pathogens. So the last thing that was really interesting for us, and it might be something that could take this to the next level, is that the Xiangming bacteria seem to be, their abundance in the tree seem to be controlled by the tree. So we have some genotypes that are really good at getting Xiangming in, and some are the less. So there is a chance that our breeders might include this trait of getting the right microbes in, getting the Xiangming in into the new cultivars. So there's a breeding potential for better use of microbiome in the future. And we have a PhD project, which I'm sort of supervising Hamish McLean, which is he's going to try to now isolate these bacteria and look at their potential and colonization and so on. So we're really excited that we have already about 50, 60 of them ready for testing, and he is progressing with that. So we hope in a couple of months to years we're going to have some good news on this front. On a completely different theme now, we have done in last 3 years, we have done some very particular trial where we looked, we know the growers were telling us that some sites have more canker than other, and if you plant later, if you store trees for very long, you tend to get more canker. So we've done a trial where we actually tested that, where we get trees supplied by FP Matthews that were grown at the same condition, inoculated to the same level of neonectria in the nursery already, and they were lifted at the same time, planted at 2 different times, one in December 18 and one in April 19. And then we analyzed the soil on the sites and looked at the canker assessments. And we wanted to know whether there will be a difference between how much cankers we will see on different sites, on different cultivars, and if the planting date affected this canker rates that we see. What we saw here, you have 3 sites, and you see the main stem cankers for the trees that were planted in April on the top, or they were planted December 18 on the bottom. And you can see that overall, it didn't matter at the end after 4 assessments, so 3 years, it didn't matter much when you planted. But the site differences are huge. So the cultivar that is the most susceptible on all sites. You can see those are the number of cankers that you're looking at each assessment. But the total number of cankers per tree is hugely different. So it goes on site 1, even the most susceptible cultivar had up to 3 cankers per tree maximum, while on the site 3, the same cultivar has almost 9 cankers per tree, and it really matters where these are. So here we're looking at the main stem cankers. So you will see in the next slide, if you have 9 main stem cankers on the tree, that tree is nearly dead or dead. So really important take-home message here, that the site differences were very, very strongly affecting the amount of cankers that we see, but within site as well. So which block within site we analyzed, it had the sort of the position within site had an effect as well. Planting had very small but significant effect, and only on peripheral cankers, not on the main stem cankers. So we looked at the tree mortality, because that's what you would be most interested in. It doesn't matter if you have 4 cankers or 6 cankers, as long as the tree is productive. But here you see that even more so, some cultivars, for example, you have Braeburn in red and Gala in orange. Those were really site dependent. So on top row, you have site 1, and then 2 and 3, and you have the columns, the 2 different colors. You have the April and December planting, and then the 2 columns that has the labeled with H represent healthy trees. The middle 2 columns, nearly dead trees, so at least half of it is dead. And then the last 2 columns are completely dead. And you can see Braeburn on site 1, all the trees are healthy, little bit nearly dead. Site 3, half of the trees are dead and half of the trees are, the other half is nearly dead. So for Braeburn, it's really important site. While on the other side of the spectrum, for something like Grenadier or Golden Delicious, they were good on all sites. So for breeding, Golden Delicious and Grenadier resistance might be quite good. It looks like it's not site dependent, while whatever Braeburn and Gala have might be very site dependent. And actually, when you look at tree mortality, planting date did not affect tree mortality much. So what drives these differences? We don't know, because you only had 3 sites and it's very difficult to compare. We analyzed the soil properties and only pH was really markedly different. It was higher pH in site 1 than in other 2 sites, but we don't know. We had very similar commercial practice, very similar climate conditions. And we, as I said, we don't have a conclusive data for soil properties. But again, Hamish will look at 2 potential candidates for these differences. One is waterlogging. Sites might have the same amount of rain, but might be waterlogged differently. We don't know. So he's looking at that. And he's going to look at the root microbiome, not just the soil properties, to see if in a root and soil microbiome, if we can find some correlations with canker. And you can read more about this in a last year's paper that Xiangming published on this, that we published together with Xiangming on this. And here I'll just try to pitch an idea that we want to take forward. Now, what we want is, rather than us setting up a very expensive trial for 3 years, we want to engage with all the growers across the whole UK, if possible. Get data on your soil properties, get data on the canker, whether you go assess 10 random trees or we come on the site, and then pull everything together if we can to get to understand these soil properties better, so you can plan better when and where to plant new orchards and how to change soil if we need to make it less canker susceptible sites. And with this, I'm going to segue into other soil microbiome amendments that we're doing for improved canker and climate change resilience in general. We know the mycorrhizal fungi are really important. They help with nutrition and water management. They increase drought and waterlogging tolerance. And importantly, they can reduce canker, like Botrytis canker that was found in India, and even neonectia canker, apple canker on cider apple in UK was reduced with mycorrhiza. We also know that trichoderma can produce or provide a degree of control, and there is a precedent for it in the trees as well. So what we're doing in this Resilient Apple Orchard project, we are testing combinations of mycorrhiza and trichoderma amendments first at planting. If you're planting new orchards, you want to know if it helps to protect against expression of canker symptoms in the first couple of years. So we have a large amount of trials here, 4 on waterlogging prone sites and 2 on drought prone sites, and we have 160 to 200 trees per site with different amendments, which we're going to get the first result this year. And we're running for 2 more years. So finger crossed, we get some good results that we can then report back. There's a lot of mature orchards as well around, which might need to increase their resilience now with more climate change and more with more canker problems. So we're trying different ways. One is get mycorrhiza in the orchards with root pruning, or by using wildflowers that will boost the presence of AMF. So the wildflowers that can be mycorrhizal, so they will increase the amount of mycorrhiza in the orchard with their roots, potentially bring mycorrhiza down to apple roots, and then sustain that and increase resilience of the apple orchard. So here we have a few trials as well that we just established this winter, and we hope that our flora margins are going to be very good in establishing, so we can have the first results then next year. So with this, I'm going to quickly conclude that we have a lot of investigation going on at the moment in terms of 2 ongoing trials for new spray products, new spray programs. We're looking at the new biocontrol in the medium to long term. And in the very short term, we're looking at how to better manage soil for resilience against other stress and against canker. So with that, I'm going to just really thank the Growing Canter Medway, Innovate UK, HGB, and BBSSC, and most important, Apple industry partners for funding and granting access to the sites, and our team for doing most of the work, and FP Matthews to give us most of the trees that we needed for the past couple of years. So thank you. And I have, no, I don't have any question time, maybe 1 or 2 questions quickly. Yeah, Scott, you muted, but
sorry, thank you. Yeah, just one question. Have rhizobacteria been tested? They're similar to mycorrhizae, but established quicker, shorter lifespan, whereas mycorrhizae takes 6 months to 2 years for full beneficial effect. Any thoughts on that?
We have tested mycorrhizal bacteria in HDB TF223 trial. Now, there is mycorrhizal bacteria and mycorrhizal bacteria. You have to have a strain that is good for colonizing apple, and we haven't had great success, we think, with the previous strains that were tested. But I think Tom in the next is going to look, they looked against apple replant. They have used bacteria, not just mycorrhiza.
And I know you've looked at plant growth promoting rhizobacteria in the past as well, haven't you?
Correct. Yeah. So that was done on apple rootstocks and on the full scale orchards, and we didn't see any positive or negative effect, but it's very difficult to gauge whether, because establishment wasn't right, because amendment wasn't right. It's very difficult to measure if they're still there after a couple of years. And maybe Xiangming can comment. I think he did some research on how long after the treatment can we still see the effect on, can we still detect those microbes? And we weren't able to detect any on our rootstocks, I believe.
Okay, we must move on. There is another question which I was also going to raise about how can
I'll try to answer in chat then.
How can growers in the industry get involved with helping you look at your site soil survey? So
that's something that we're going to try to first get some funding to do the full scale thing, and then we will contact through our industry.
I can help with that.
And through Scott, we're just going to try to cast as wide as net as possible, really.
Yeah. Okay. There is another question coming. I am going to have to move on, but maybe Matt, you can have a look at the chat facility and reply to all there for that question for you there. So, but for now, thank you so much, Matt. We better move on. So our second speaker for this session is Tom Passey. Tom is a pathology colleague of Matt's at NIAB at East Malling, and Tom has been doing some work on apple replant and soil amendments. Good afternoon, Tom.
Afternoon, Scott.
That's fine. Do you want to hit your slideshow?
I'm hoping there it is. Can you see that?
Go. That's perfect.
Perfect. Right. Okay. So yeah, I'll be kind of looking at a different issue to Matt, but some similar themes going throughout. So which we'll crack on. So yeah, I'm talking about apple replant disease. So like Matt, I'll quickly cover what we're looking at, although not quite as clear as it is with apple canker, where you've got one clear fungal pathogen causing the disease. With apple replant, we don't have that. So what apple replant is, it's the poor establishment of young trees, where you have apple going back into land on quick rotation without any change in crop or period of a fallow period between putting the apples in. And when the new trees go in, you tend to see issues with the establishment and growth of those trees, with vigor, branching, and yield above ground, and below ground with root mass. And you tend to, if you take the roots up, you tend to see necrosis of the roots on the root tips. Most of the trees will survive, but as I say, you have this reduction in this reduction in yield that can be sort of 2 to 3 years later in producing fruit to start with, and then can be 50 to 60% less yield. Also, when severe, the young trees can just die and never get to yielding anyway. So as I say, it's not as easy as with canker in terms of what causes it. There's been decades of discussion about, is it to do with physical properties of the soil and various different aspects, and it turns out it's sort of a complicated complex, as I put there. So the causal agents are highly thought of to be now sort of fungi, myocetes, bacteria, nematodes. But what the important aspect is that's causing the replant can be very variable from site to site. And so researchers in different countries and in different areas have often come up with ideas of what causes replant and dismissed other ideas, but what is the consensus now is that it is so variable. And what we see with apple replant disease is that it's not associated with large changes in the soil community, but it is just a number of specific microbes that are either higher or lower in one site compared to another, which then has a big effect on the replant. So some of the candidate pathogens there, Fusarium and Rhizoctonia, some of the Phytophoras. These are the species that we think are the main players when it comes to seeing increased pathogens when you look to see what it is in the soils that's causing the replant. But also it has been shown where you have a decrease in some of the beneficial microbes we see. So we've just been talking about AMF and PGPRs, and actually it could be that some of the soils that are struggling don't have as many of those beneficial microorganisms. Tricky to do much about, as we don't have a huge number of effective broad spectrum fumigants that are allowed to be used anymore in the UK. So the main rider that we look at is using rootstock genotypes that show a resistance or are less susceptible to a replant. So I move on to some work that we've done previously. A couple of years ago now, we were doing a UKRI Happy Cool project where we set up a orchard in Worcestershire, one of the Bulmers Heineken orchards over in Worcestershire, where we were looking at 7 different rootstocks with the Scion Worcester on top. And we planted the trees straight into holes that had trees in the previous growing season. And also we planted exactly the same genotype trees in the alleyway between the old tree stations. And we wanted to have a look and see how the trees established, how they grew. And we did this throughout the Happy project. And then Chris Cook, as part of his CTP PhD studentship, carried this work on. So we had a few more years worth of data to look at afterwards. So you can see here the trees on the right and the trees that were planted into the tree station. So as I say, they've gone straight into the ground where the trees were previously. The trees on the left of this photo are the trees that went into the grass aisle. And it's fairly clear to see that there's definitely a difference between the 2 different trees, and there was a strong replant effect on this site. So much so that we've got this figure here, where you can see so these are the different rootstock types that we had in the orchard. And we've had a look at how the tree growth compares between the trees in the tree station and the alleyway. And you can see here that the bottom 2 lines, the blue and the red line, are showing there's no replant effect there with those genotypes. And then we have sort of 3 that are shooting off at the top there. M116, MM106 and M200, which on this site showed the most severe replant. So this is where the trees in the tree stations were much smaller. The growth was smaller compared to that of the same trees in the alleyway. So to show this point further, this time the different rootstocks have been given a replant disease score. And it was interesting to see that where going into the trial, we were expecting M116 and MM106 to be two of the more tolerant genotypes, rootstock genotypes towards replant disease. But actually what we found was in this trial, they were the two most susceptible. And what was interesting was that the trees that were planted on that site that had just been grubbed were previously on MM106. And MM106 is also a parent of M116. So what we've sort of theorized from that is that actually what we tend to know about rootstock susceptibility might not necessarily be true. What's important is knowing the rootstock that the trees were on previously and the heritage of that rootstock. The parentage is potentially important, and the parentage of the genotypes of the rootstocks that you're planning on then using in the following seasons. If you can vary between the different genotypes of rootstock you're putting in, it might be useful in terms of getting over this replant disease issue. So within the work, we also had a look at what's happening in the ground. We want to know what's happening with the microbial communities within the soils of these trees. And what we found was there is a strong grouping of the fungal and bacterial populations in the rhizosphere soil. So this is the soil that is immediately around and in contact with the root. And what we found was you can see sort of the groupings in the circles on these two figures of the fungi and the bacteria, where the triangles and circles, the triangles showing the tree stations and the circles showing the alley trees, the community profiles of what's living in the soil closest to the root is quite different between those two groups. Going into having a look at what actually some of those things are is quite tricky at the moment with the sequencing we do. So this is where we've extracted DNA, sequenced what's living in the soil that we've sampled, and try from that to work out what it is, what some of these causal agents might be, what the where we're talking about the various different issues that are causing ARD, what some of those might be. It's quite tricky to do with the power of the sequencing that we're using at the moment to really get down to sort of genus or species level. But we have pulled out, and you can see from the table here, we have pulled out a few potential causal fungal agents, sort of, and also interestingly, a couple of beneficial differences in beneficials, where there are a couple of beneficial fungi associated with AMF. So again, differences there showing that it can be both increase of pathogenic and decrease of beneficial microbes. So that moves me on to our current research, which is based around a EU Horizon 2020 project called Excalibur. This is the website, and there's plenty of information on that website about the whole project, which is quite an overreaching project. There's a lot of different trials going on across Europe around this project. The idea is to enhance the knowledge of what's happening in the soil, the dynamics and the synergistic effects of some of the probiotic microbes that are either already in the soil or we can add to the soil, and how that can affect our horticulture. The specific objectives to identify and define how soil management strategies impact on the inocular and what's already in the soil, but also to develop new byproducts that will hopefully aid the trees in various aspects around horticulture. So the model organisms being used in the project are apple, strawberry and tomato. There's 16 partners across 11 countries in the project. So the work we're specifically doing at NIAB is based around apple replant disease, as you may guess. And we've been running a number of trials across the past couple of years. Most of these trials are ongoing, and I'll just run through a couple of them now. So the first trial that I'm talking about is where we've done a potted trial. So we potted rootstocks, M9 rootstocks, 1-year-olds, in pots in a polytunnel. This was done in 2021. And half the trees we assessed after one growing season, so in autumn 21, and then the second half of the trees were left for 2 years and were assessed in the autumn just gone. So in this trial, we've been looking at using different amendments of various types, both individually and together. So that some of these have been fungal organisms, so trichodermas and AMF, and some of them have been bacterial, so Pseudomonas, Bacillus. And we've been looking at how we've been comparing them used on their own and comparing to whether they work together. Because of it being such a complex disease where we don't know quite what might be the cause of replant disease, it might be that different beneficial amendments work better against some of the causes than others. And actually, it may be that a combination of some of these beneficial organisms works better than just relying on one or two on their own, especially if you're trying to roll this sort of thing out to various growers as opposed to factoring on just one particular site. So what do we find? Well, after the first year, not a huge amount. There's not a huge great difference between the height or the girth measurement. So we measured girth of each of the trees before and after the growing season. And there doesn't seem to be a huge amount of difference seen in that work. The second year data hasn't yet been analysed, so we'll see actually if there's any change after 2 years growth. So we'll see whether it just takes a bit of time to see those differences or not. And then we also looked at the soil communities for the fungal communities in these pots. There wasn't a huge difference, but with the bacteria, there was the main differences was where we either sterilized the soil. So this was sterilized in an autoclave to hopefully kill off anything really that was in the soil at planting. And then the other product was a biofumigant product, or a product that can be used as a biofumigant. We found that the amount we used in a small pot wasn't great for then growing trees. We've used it in other trials in the past in the field and never had any problems, but when it's in a contained area, it obviously has a greater effect on everything that's going on there. The other trials, then we're running our field trials for this project, looking again at the use of individual or a combination of different beneficial microbes. This orchard was planted in April 2020, and we are actually one of the things we are looking at is how much some of the trees we are inoculating, re-inoculating. So we re-inoculated last spring, and we will re-inoculate this spring. And what we're interested to see is we will compare how the trees that have been re-inoculated compare to those that were only treated at planting, and see if you do need to keep reapplying some of these beneficials to really have an effect going through. That I should note that we haven't done that with AMF because it was going to be too tricky to do for individual trees, where we just have plots of an individual tree. We have a number of them across the trial plot, but it was going to be too tricky to do, and we didn't want to damage the roots of the newly establishing trees more so than we needed to. But we have added the other organisms that we can apply as a drench. The other trial we're running then is kind of moving on from the Happy project that I talked about earlier, where we're looking at different rootstocks with 2 different Sion cultivars, and we're looking at we've planted, as you can see from the image on the right, we've planted in the old tree stations and in the grass soil between the trees that were previously in that orchard. This was planted in October 2020. We have had a quite a large canker problem in that orchard. So kind of going on from what Matt was talking about earlier, it will be interesting for us to also, although that wasn't a primary aim of the project, it'll be interesting to see if we see any canker differences, and whether there's a kind of link between replant and the beneficials and canker development as well, where we've seen such a site-specific issue with canker. Is there a link of some sort with replant there as well? And some of the assessments we're doing for these trials, we're looking at yield, flowering time and intensity, girth measurements for trunk growth. And then we're also doing annual assessments of what's happening in the rhizosphere. So on these roots, that means digging down to the base of the tree, finding some roots, pulling them out, and then extracting the DNA and see if we can find any of the beneficials we've put in, but also have a look and see how the community, each tree is changing from planting across time, and see whether there's any differences in what's there, whether they be pathogenic or beneficials or anything else in between. I should just say that, so this is an ongoing project. It's a big European project, and most of that data is being analysed by our partners. So I haven't got anything to present now, but hopefully we've got another year of the field trial to run. So hopefully by this event next year, or certainly in 2 years' time, we'll have all of the data from this project, and we'll be able to really show you what we found within it. Just thanks to funders and a number of colleagues there, past and present, especially Chris Cook, who recently finished his PhD and has now moved out to Washington State in the States and is carrying on work on replant. So it's good to see. But a lot of the work presented here is work that he was doing in his PhD as well.
Thank you, Tom. We don't have any time, I'm afraid. We're running over at the moment, but thank you so much. For people's information, there are a couple of comments on the chat facility about some of the rootstocks and apple replant. Anybody got any further questions, please direct them to Matt, to Tom. Thank you, Tom. And we're going to move straight into our final presentation of this session, which is Xiangming Xu, who again is known to most of you, who's the Head of Science at NIAB and at East Malling. Good afternoon, Xiangming.
Good afternoon. Hope everyone can see my screen.
Thank you, Xiangming. I don't know whether you can do this in 10 minutes, can you?
Okay, I can do it very quickly. Okay.
Fantastic.
Okay, good afternoon. We talk about canker, but this is bacterial canker for cherry. And also this project is funded by BBSRC. It's quite a fundamental science. But what are I trying to do? Trying to really give you an idea why we're doing it, how the results from this fundamental research could affect the practical disease management in 5 or 10 years' time. So that's my reason. Okay, the next slide. Essentially, we're trying to show it's the Pseudomonas syringae, a PS, is causing cherry canker. And it has a really interesting annual dynamics. So in the spring, the Pseudomonas move from the canker on the shoots onto the leaf surface as epiphytes in the summer. And on some varieties, it may cause a short haul symptom or some infection on fruit. But generally speaking, most remain symptomless on leaf as epiphyte. Then in the autumn, they move back to the shoots again. So the whole idea of this project really is looking at in the summer phase when the Pseudomonas move from canker to leaf surface, look at the epiphyte, how do their population change in the summer, how that relate to the disease development in the autumn. So the question we are asking is how the PS epiphytes on leaf surface really change with time and with space. Particularly in relation to the cultivated cherry orchard or in wild woodland. And essentially we're asking, is the population or the potential pathogenicity of the PS changing with the environment or changing with the host or with different, also whether the population change with our conventional fertigation scheme or different management practice. So first question is we're asking, is the PS influenced by environment? So we have 4 regions: Scotland, Hereford, Southwest, and Kent. In each region, we select several orchards and several woodland, and also we have several cultivars, several trees in each cultivar, in each orchard. Then we sample the PS, the leaf from the leaf surface, and then we use the sequencing technology to understand population dynamics. Similarly, we sample in each region, in several sites. We sample the leaf surface sample from 5 or 6 different related species in the woodland, and try to quantify the PS level on this host. What do we find out very interesting, is through molecular sequencing, we showed the potentially pathogenic Pseudomonas syringae as a proportion of the isolate we obtained is far higher in Kent and Southwest, particularly in orchard. In Scotland, virtually 0. So the question we are asking, is that because the total population is so low in Scotland, so we couldn't get many isolate out, or maybe simply because the proportion of isolate with potential pathogenic in Scotland is so low because of temperature? So that's one of the key questions we got to try to answer, get further funding to answer that question. That's really had direct relevance to the cherry production in different regions. Next question we are asking is, how does the cherry canker population change with covering plants or with nitrogen input? So this work has been done at Xiangming, and we will get data hopefully by September this month. So this will be really interesting to see whether population size and the proportion of potentially pathogenic PS change with our cultural practice. Next thing is very interesting is, you all know, once the PS move from canker to leaf surface looks apparently healthy, but there are so many other microorganisms on the leaf surface, we call it all epiphytes. The question we're really asking is, if some isolate of PS is non-pathogenic, some is, could the pathogenic isolate pass the virulence factor to the non-pathogenic, make those previously non-pathogenic become pathogenic? So that's the question we are asking, is, does the evolution happen on leaf surface so quickly you can pass the pathogenicity factor from one strain to the other strain? Okay. So first thing is first, it's very interesting. We look at the isolate from same region or same plants. You can find so much variability in virulence among the natural PS isolate. So essentially, on a single leaf surface, is the ability of PS to infect plants is not uniform, it's so variable. Then that gives us a question: could that variability contribute to the movement of pathogenicity from one strain to the other? So to do this, we really looking at one of the common potential way of moving pathogen moving between strains is to the phage, which is a bacterial virus. Okay, so look at the imprint or footprint of phage, we could infer whether the actual pathogenicity effect can pass between strains. So far, we obtained clear evidence, the molecular evidence, to show the virulence factor can be transferred from a known pathogen to a previously non-pathogenic strain or epiphytes. Okay, so that's again. So finally, and it was more interesting, is suppose nowadays the sequence is so cheap, genomic sequence. We also know not every isolate of PS is pathogenic to specific hosts. That question we ask: can you predict using genomic information to say whether that particular isolate can infect in cherry or any other particular species? So this one, we're trying to use the machine learning AI to use published information on the sequence information plus the known host, and also new information generated in this project, really to developing AI algorithm to predict host range from genomic information. Okay, so this is what we're trying to do. The question really would say, okay, how does the work affect the particular commercial production of cherry? So these are potential questions that could be impacted by the current research result and research outcome, although it's a very fundamental research. First one is really, question is, when you plant a cherry in a particular region, the question always is, what is the inoculant availability in relation to the environment, to the woodland species available at that particular site or region? And next thing is, can you use common agronomic practice to reduce the inoculant availability, or even to disrupt the evolution or annual cycle of the cherry canker pathogens? And equally related to the third question is, if the complexity of epiphytes influence the evolution ability of the community to acquire the virulence factor, is there any other way using agronomic practice we can disrupt that evolution or reduce the rate or minimize the rate of evolution of the epiphyte acquiring virulence factor? And finally, it's obvious, if you want to plant a new orchard in particular site, the question is, you could ask, can we use any information on the Pseudomonas swingi isolate in the region to predict whether we have any isolate that can infect cherry? So those are, although this question cannot be answered now, but it's very long term, but it's really fundamental, very important for commercial cherry production in the UK, given cherry is such a high value crop. So final slides, this project is a huge project funded by BBSRC, NIAB, and also Scotland, I think, office. The collaboration between Birmingham, University of Birmingham and Imperial College, and also University of Reading, and the most work is done at Cambridge group, but it's more really focused on helping them do the management trial on the fertigation and covering crop. So I'll stop here.
Thank you, Xiangming, and you've won us back some time. Very grateful to you for that. Just question for me is, what's the length of this project? How much longer does it last?
We have one more year to go.
Okay, so hopefully